Welcome to the Certara Drug Interaction Solutions video, presenting the clinical pharmacogenetics dataset in the Drug Interaction Database, or DIDB. The clinical pharmacogenetics dataset contains study results with in-depth analysis of the impact of genetic variants of enzymes and transporters on the PK, PD, and safety of drugs in various populations. Let's look at the drug clopidogrel, a platelet inhibitor indicated for the prevention of myocardial infarction and stroke. If you have the citation accession number of the study you want to find, you can search for it by clicking on the search icon on the upper right-hand corner of the page and typing it in the search bar that appears. Once you're on the citation page, this hyperlink right here will take you directly to the source webpage for this citation, which is PubMed in this case. The box on the left-hand side indicates that this study contains clopidogrel PK and PD data associated with CYP2C19 variants. Side effect data are also curated if available. Clicking on the study number on the left menu or scrolling down allows you to view the data in a table format, starting with the study design and drug administration, the genotyping method used to identify the alleles tested, which are listed on the right. The table starts with full descriptions of each study population. The populations with impaired phenotypes, here intermediate and poor metabolizers, are tested against the group harboring the wild-type genotype or normal metabolizer phenotype, which is always designated as the reference group. All PK parameters measured for the prodrug clopidogrel, which is bioactivated mostly via CYP2C19 to its active metabolite, clopidogrel thiol metabolite H4, are displayed here with their respective percent changes versus reference group. The impact of variant is based on the statistical analysis performed by the authors on percent change in AUC, clearance, and concentration, and/or by the author's conclusion. In this study, a gene dose effect is apparent with progressive accumulation of clopidogrel exposure in the CYP2C19 intermediate and poor metabolizers. As expected, this is inversely correlated with a progressive reduction in H4 exposure. In addition, for the polymorphic genes and compounds that are studied, useful resources like NCBI Gene, PharmGKB, PharmVar, and PubChem are provided when available. By clicking these hyperlinks, you can quickly find a summary of their description from these open resources. When available, PD evaluation is also curated and displayed here. In this study, parameters associated with the inhibition of platelet aggregation are compared between the three CYP2C19 phenotypes. If you want to find out what other drugs are significantly affected by CYP2C19 polymorphism, you can do so under Pharmacogenetics queries. Click on Queries from the top menu, and then click on Pharmacogenetics. Once you're in, select the tab Search for Quantitative Results. Under Genes, enter CYP2C19 and select. For our specific search, we leave the fields Compounds and Populations blank since we want all compounds regardless of population type. Let's hit Submit. This opens a new page with the results presented in a table. Of note, additional measurements which are not involved in the determination of the overall impact are currently hidden. I'd like to invite you to view the document describing how we determine the overall impact of a pharmacogenetics study. You can customize this table according to your preference. A detailed video tutorial is available by clicking on the Learn More About Our Table Features button. For example, you can hide the columns you don't need and reorder the columns by dragging and dropping the column header. If you want to order the results by drugs whose AUC is most significantly affected by CYP2C19 variants, find the AUC percent change column and click the sort icon next to it. This allows you to order the results in ascending or descending values. Note that you may also select a range of exposure. Let's say you want to see the results for CYP2C19 POR metabolizers. Type the word POR under phenotype to filter the results. Once you are done, you can copy your customized table to the clipboard, download it as an Excel or CSV file, or print it. And that concludes this video on the pharmacogenetics dataset. I invite you to check out our other video tutorials under Resources, which provide an in-depth look into the contents and functionalities of the DIDB. If you have any questions, just go to our website, druginteractionsolutions.org, and click Contact Us from the top menu or on the right side of the screen. Thank you for your time.